Thank you very much. I am not going to be very long because I think you had already a lot of information so far, and I thought that given the context, I would go back to an example, a concrete example of what the resources that we have can do. And so I prepared some work. So we're working with a group which is called Viral Zone. So Viral Zone is a resource you can find on Ecstasy as well, and it's actually a real encyclopedia on viruses that is connecting to sequence databases. And they have, if you don't know this resource, I really recommend it to get, as I said, the encyclopedia information on viruses. So we actually, all of us at SIB, if we have any connection with COVID-19, so Viral Zone is certainly on the forefront with the SARS-CoV-2, CoV-2 COVID-19 coronavirus. So this is the news feed on the resource. As I said to some of in the practicals yesterday, we have also included a lot of data, everything that actually was released on the web regarding the spike protein, because this is the only glycoprotein in GlyConnect. So I made a point. So you see that we have, since yesterday, now you're a bit familiar with our little color code for the information that is included in the database. And so you can see that in each case, Glygen mentioned yesterday that they have included this data set. So this is actually the only purified native-like protein, where all the others are commercially available. However, all of them are recombinant. They have been expressed in expression systems. So the most used is the HEC 293 cell line, but there's a derivative of that cell line. This is an insect cell line. So all sorts of different expression systems have been used. So two references have used the HEC 293, one only with subunit 1, the other one with the 2 subunits. You can see that there's a lot of composition associated with them, and different figures each time. And so I just didn't take the time to look at this data, because I actually give a talk on that. If you're interested, it's a little bit of self-advertising. I apologize, but still, I didn't want to repeat myself. So I go back to Viral Zone, and the work we're doing with them is to actually look at the interaction and cross-reference Viral Zone with what we have on host pathogen interaction, and this is in the SugarBind database. And so this is the cross-link. So in Viral Zone, we have 92 results on influenza virus binding to human, so host sugars. And so this is the database itself. This is, it's actually developed by Julien Marietto. And we have a lot of information. You see 92 results don't fit into one page. So I'm talking about here, for instance, one example. Each time you mouse over, you've seen that with yesterday when I mentioned GlyConnect, that you mouse over these names, you have the SNFG code. Sometimes you have a name. And sometimes when we have affinity data like glycan array data from the CFG, most particularly, we are reporting this information here. So we have a similar code as we have with GlyConnect. We're trying to be somehow consistent. So we have the tissue here, the disease, and the protein are only lectins in that case. And if you click on the structure, you have a page structure with the references and some information with the pathogens that are involved. So if I focus here, all the lectins, in fact, they're not different lectins, and they're only different strains of influenza virus in that particular case with different tissues. So this is the flu as a disease, and we can actually connect to glyconnect with a substructure search. So you can see here that all of these larger structures from glyconnect contain the ligand I just showed before. And so if you click on one particular here, we go back to glyconnect, and you can have information on the protein. So here it's a mucin, and on the tissue, and here it's respiratory mucosa. We've actually updated, as you saw yesterday, I mentioned that we have now connected everything to the Uberon ontology. So the information here is such that you can actually connect from sugarbind to glyconnect via the substructure search and see that there are possibly some glycan-mediated protein-protein interaction between the virus, so the outside protein, the surface protein of the virus, which is actually recognizing a glycan sitting at the surface of the host cell, and this is described in glyconnect. So I did not mean to really spend that much more time in developing. We'll have some more details on sugarbind this afternoon. It's not only limited to viruses, but it also talks about pathogen bacteria. And this is somehow the landscape as we try to characterize it between viral zone and what we're doing. You heard about unilectin a bit earlier, so there's more about bacterial lectin and viral lectin in unilectin. The real binding experiments reported in sugarbind. We have, of course, the glycoproteins in UniProt and NextProt when it's human that are also described. So glyconnect, you have heard now about it, and this is all somehow developed at SIB. And again, a little bit of self-advertising. We have explained this in a short video with Philippe Lemercier, who is the head of Viral Zone, and myself too, and these are some of the part of the slides I showed is in that video. So I did not want to spend that much more time. Viral Zone is and our group are really trying to collaborate so that there is a bit more recognition about the role of glycans in the host pathogen interaction. I still have our bridging advisor from UniProt, Elizabeth Gestiger, that I always acknowledge, and our funding. So this is all I wanted to add for today regarding a particularly opportunistic example of what we're doing with different resources and focusing on glycovirology.